Hi, he's right up here. I'm gonna drop this at the front.
Okay, so we're going to finish up with the last lecture we were doing on mitochondria. Before I will get into it, though, just a brief announcement about the test. Most of you took it. Most of you did very well, surprisingly well, suspiciously well, and we hope to post the grades soon. Just as usual, Brightspace, some learning curve for us, so we want to make sure everything is in order before we post it, and in case it disappears, so we're backing up stuff. So, but just quickly looking through, most people did very well. We're probably looking at about a 90-ish average, so it's very high. Okay, so we're going to continue on and talk about mitochondria. The last slide, I think we were talking about fission and fusion. So the idea that mitochondria obviously can join together as fusion, they can break apart fission, and there's a number of proteins that are involved in that. The mitofusion is MOPA1 for fusion and DRP1 for fission. So another interesting aspect that a lot of people don't appreciate about mitochondria is they actually can be a storehouse for the ion calcium. So calcium actually plays a really important role in many biological processes. It can be imported, it can be exported. One of the major places of storing it is in the endoplasmic reticulum, which we'll talk about in a few lectures. But another major place is the mitochondria. And so it's a charged ion, so it can't cross membranes, but it can go through the outer mitochondrial membrane via VDAC, porin that we talked about last lecture. And to get through the inner mitochondrial membrane, there's a protein called the mitochondrial calcium uniporter. It's a one-way ticket goes in this way. So when calcium is imported at a moderate level, it can actually bind to and activate a number of enzymes in the tricarboxylic acid cycle or citric acid cycle or Krebs cycle, pick your flavor. So it can actually promote increased mitochondrial activity through the Krebs cycle, which ultimately will produce NADH, which helps drive the electron transport chain, and ultimately through an oxidative process is going to generate ATP, which we talked about last lecture. However, if there's a dramatic increase in calcium intake, it can cause problems for a variety of reasons that I won't get into, but it can actually trigger the formation of this thing called the mitochondrial permeability transition program. Now, I'm going to talk in a lot more detail towards the back end of the course, so don't worry about all the details of what that is right now, but just know that release of stuff from the matrix or the inner membrane space into the cytosol can trigger programmed cell death. And calcium can, either its rapid efflux into the cytosol, or its triggering of the opening of this pore and the release of other molecules. We'll talk about cytochrome C in more detail as an initiator of apoptosis when it's released into the cytosol. So high levels of calcium and cytochrome C, so calcium in the red dots, cytochrome C in the blue dots, can actually trigger apoptosis through some complex mechanisms which we're not going to talk about right now. So it's just important to note that calcium plays a major role in mitochondrial function, plays a lot of beneficial roles, but too much can actually be harmful. It can actually promote the mitochondria to release factors which can trigger the death of the cell, via programmed cell death or apoptosis. All right, so this is sort of hot off the press. This is released, a paper that was published last month in the journal Science. And so, how many people here know what the concept of a synapse is, a neuronal synapse? Okay, most. So the idea is that neurons talk to each other. So you can have what's called a presynaptic neuron, and it'll have an axon. You don't see it here, but it'll be a long thing. And at the end of that axon is a little swelling, a bouton. And so it can act on an adjacent neuron. So this is the postsynaptic neuron. It doesn't do it directly, but there's a little space, it's called the synaptic cleft. So these points of close contact, not direct, but they're close, where you have an axonal boson and then a synaptic structure here, postsynaptic structure, is that when the neuron fires here, it releases a neurotransmitter, and it's not shown here, but it'll trigger activation of neurotransmitter receptors. And that, in many cases, can cause an opening of a channel to import calcium. So calcium goes from the outside of the cell to the inside of the cell. Calcium can then, once it accumulates inside the cell, be transported, as we just talked about through those mechanisms in the last slide, into the matrix of the mitochondria. And there, you can do a number of things. So I talked about last slide that it can actually bind to and activate some of the Krebs cycle enzymes, but it also can do something interesting, is that it can activate certain kinases. These are proteins that put phosphate groups on other proteins. When we do the signal transduction lecture, we'll talk about that a lot. But these kinases, when it's activated by calcium, so calcium can bind to that kinase, and that will phosphorylate a transcription factor. It's not important which transcription factor it is right now. But this transcription factor, which is found in the mitochondrial matrix, can bind to mitochondrial DNA and promote the transcription of a number of mitochondrial genes. As we talked about last lecture, that the mitochondria has its own genome and codes for a lot of different things. One of the major things that it codes for are components of the electron transport chain complex. And so by increasing mitochondrial transcription, you can cause the increase in synthesis of mitochondrial electron transport chain proteins, which can integrate into the mitochondrial inner membrane and increase electron transport chain activity, increase ATP production. So what they showed is, in this paper, is that calcium is driving this. So when we have the firing of a neuron and it affects the downstream neuron, it'll cause activation of a number of things, including calcium influx, which goes into the cytosol and ultimately into the mitochondria, and will increase, result in increased transcription of mitochondrial genes, which will promote increased oxidative phosphorylation and make high levels of ATP. Why is that important for memory? Well, memory is, in a very kind of crude, broad brushstroke description, is the strengthening of synaptic connections. So what does that mean by strengthening? It means when one neuron fires, a presynaptic neuron can readily trigger the activation of the postsynaptic neuron without having to put too much energy into it. So it's basically causing this postsynaptic neuron to respond to a very low amount of stimulus from the presynaptic neuron. That relies on gene expression changes. It relies on structural changes to the synapse itself. So you make more synapses. So all of those processes require ATP. And there's this old saying that neurons that fire together wire together. Well, that's kind of the underlying concept of memory. Is the more that this fires, you have this firing here of the presynaptic neuron and response by the postsynaptic neuron, it basically sets up a number of gene expression and actual structural changes that lead to the strengthening of the synapse in the sense that you need very little neurotransmitter release here to elicit a firing of this postsynaptic neuron. And that can lead to the formation, and this is very crude, broad brushstrokes, of new memories. So that's all fine, but what happens when we get older? And as we talked about in the last slide, is that as we get older, our mitochondria don't work so well. They don't work so well for many reasons. Mitochondrial fission and fusion doesn't work as well. Mitochondrial DNA replication doesn't work as well. You get increased mutations in mitochondrial DNA. So all of these things collectively lead to less mitochondria found within neurons that are able to make ATP as readily. And so what they did is to kind of say, well, what would happen? So this is dependent on calcium increase, and calcium import is also another thing that tends to decline with age. So all of these things are working against us. Age screws up so many things that it's kind of difficult to figure out what part are we going to try to fix, particularly when it comes to our brains functioning as we get older. They don't work so well. So in this study, what they did is they genetically engineered the transcription factor to be turned on all the time. It didn't require the presence of calcium. And as a result, this was able to increase mitochondrial transcription of those electron transport chain complexes, and it was able to reduce or sort of partially rescue that age-dependent decline in ATP production, because mitochondria are just not making those mitochondrial transcripts. So we can kind of in a way biohack the aging process, at least for memory, to artificially increase the transcription factor activity that doesn't rely on the calcium. And that would lead to increased transcription of mitochondrial DNA and could partially rescue some of these age-dependent decline that we see in memory. So kind of interesting. And the basic concepts that we learn in cell biology are applied to major medical discoveries all the time. Okay, so that finishes that lecture, and now we're just going to move on to the endoplasmic reticulum and protein translation. Okay, not showing here. Great. Now it is. Okay. Okay, so we're going to talk about how, we got a lot of proteins that are made in our cell, and they have to figure out where to go. And we'll talk about the concept of protein targeting. We'll talk about targeting proteins that have to go into the endoplasmic reticulum. Why is that important? Well, certain proteins need to go to things like lysosomes, or they need to be secreted, and they're going to have to go through the endoplasmic reticulum in order to do that. How do proteins get inside the ER? And we'll talk about co-translational translocation, talk a little bit about how proteins get modified in the ER, and then briefly we'll also talk about how proteins are targeted to mitochondria and nuclei. Okay, so in a typical mammalian cell, you can easily have like 10,000 different proteins being made at any particular time. So they have to go to the right location. And so we have to find some way to direct them in a controlled manner to their proper location. That's where the idea of targeting comes into play. So that there has to be some system within the cell to recognize that, hey, this is a mitochondrial protein, this is a nuclear protein, or this is a protein that has to go to the ER. So we need to initially target those proteins to the right destination, particularly if it's a certain type of organelle. And we'll talk about that some proteins are completely made in the cytosol, and then they can be targeted, or some proteins have to be synthesized or partially synthesized, and they're targeted. And we'll explain that in a minute. Sorting generally refers to proteins that are first synthesized in the endoplasmic reticulum, and then they're moved to other organelles. We'll talk about the Golgi apparatus, and we'll talk about lysosomes. Or it could also be proteins that end up going outside of the cell or secreted. So sorting is, when we talk about sorting, it's usually referring to proteins that start off in the endoplasmic reticulum, and they're going to go a number of different ways. They can either go to the Golgi, they can go to the lysosomes, or they can be secreted. That's part of the sorting pathway. And as I go through these next lecture slides, I'll make it clear what I'm talking about when I say targeting versus sorting. So many proteins are simply synthesized in the cytosol using cytosolic ribosomes. So we should all be familiar with protein translation, but how messenger RNA is assembled, transfer tRNAs come in, that helps link, that are associated with amino acids, and the amino acids are linked together to form a polypeptide. That's basic high school biology and also first year, either biochemistry or biology. So I'm not going to go over that. So proteins that are made predominantly in the cytosol can be targeted to organelles such as the endoplasmic reticulum, the mitochondria, plants, chloroplasts. We didn't get around to talk about peroxisomes, maybe next year we will, or the nucleus. So they usually have what's called a signal sequence that helps target them to those destinations. In other cases, we have proteins that can be synthesized on ribosomes that are attached to the endoplasmic reticulum, which I'll talk about in a little bit more detail in the next slide. So these are proteins that we want to get into the endoplasmic reticulum, and then once they get in there, then they can be sorted to either the Golgi, the lysosomes, or the plasma membrane. Either it can be inserted into the plasma membrane or simply secreted and spat out of the cell. And when we're talking about sorting, generally we're talking about either non-secretory, so these are proteins that may reside in the ER, and they may go back and forth between the ER and the Golgi, or they end up going to the lysosomes. So that's the non-secretory pathway, and the secretory pathway are obviously the ones that go to the plasma membrane or are ultimately secreted to get to the outside of cells. And we'll go into, don't worry, we'll go into a lot of detail on all these steps in just a minute. So, as I mentioned, here's a little bit more visual to explain this. We have a protein that's synthesized in the cytosol. So we have cytosolic ribosomes that's reading the messenger RNA. And when that protein is synthesized, it usually has a targeting sequence on one end. Usually the amino terminus, sometimes the carboxyl terminus. It varies depending on the protein. And so that signal sequence can be recognized by quite frequently other proteins that can help guide it towards either the mitochondria or to the nucleus, or chloroplast if it's a plant, or peroxisome. So these targeting signal sequences are really important for taking a fully synthesized cytosolic protein and directing it towards the proper organelle. Now, if it's a protein that we ultimately want to be sorted in the ER and Golgi or secreted, it's going to have to get into the endoplasmic reticulum. And so that creates a little bit of a challenge, which we'll talk about in the next couple of slides. And so what usually happens is the messenger RNA, so that red line is messenger RNA, will bind to ribosomes associated on the endoplasmic reticulum. And then it will start translation. So there may be an initial bit of translation that starts to occur and on a free-floating ribosome. But then this ribosome will then ultimately move towards the ER. And we'll explain the steps in a lot more detail in a few slides. Okay, so that's sort of just the general idea, is that we have signal sequences. Some of them are targeting signal sequences that guide cytosolic proteins to the mitochondria, peroxisome, nucleus, or chloroplast. And then there's other signal sequences that can guide an emerging polypeptide towards the endoplasmic reticulum membrane. And usually, if we have to get a protein across membranes, you need a channel. So it could be what we'll call a translocation channel, or import type of protein, which we'll talk about in mitochondria. Proteins don't just cross membranes. They need help. It needs something that, like a hole, so to speak. It's usually a protein-like structure. We talked about, for example, mitochondrial VDAC. Okay, that's fine for smaller molecules, but if we need bigger molecules like proteins, we need special types of channels, which we'll talk about in a minute. Okay, so the endoplasmic reticulum is basically an extension of the nuclear membrane, and it's uninterrupted tubules, and so it tends to be found around the nucleus. It spreads out a little bit, and when you see all these dots, those are ribosomes. That's what we call the rough endoplasmic reticulum. And so the other thing to take note of is, I think Dr. Pemberton will cover some aspects of electron microscopy, so I'm not going to go into that here. But these black and white images were taken of cells using transmission electron microscopy. It provides the highest level of detail that you can see inside of the cell, and you tend to see these black and white images. And so what this is showing is that that's the endoplasmic reticulum membrane, and those little balls are actually ribosomes. And as I said, the endoplasmic reticulum extends from the nuclear membrane, and these tubules are also referred to as cisternae, and they're sort of stacked on top of one another. And then we're going to talk about some of how these ribosomes associate in the endoplasmic reticulum, and how that's important for guiding emerging proteins that are just starting to be translated to go through a translocon and enter into the lumen of the endoplasmic reticulum. So in the middle there's a lumen, that's our tube. You have the inside of the tube, and the tube walls are membranes, composed of membranes, and associated with ribosomes. So this is kind of a cartoon rendition of an experiment that happened many, many years ago. And so imagine if you go back into the 60s, people started to understand a little bit about cells, and they saw these structures, and they saw the endoplasmic reticulum. But the question was, how do proteins get from the inside of the cell to the outside? And so these types of proteins that are secreted, the theory, a hypothesis was that they must end up going through the endoplasmic reticulum. There was some evidence suggesting they did, but nobody knew 100% certain if that's what happens. So what they did is an experiment, and they took cells and they homogenized them. That's a way of breaking them up. And when they break them up, particularly the endoplasmic reticulum, it kind of reforms these small little vesicles called microsomes. So a microsome is sort of like a representation of the endoplasmic reticulum. When you lyse a cell, when you kind of break it up into pieces, they spontaneously form these little vesicles called microsomes. And the microsomes resemble in some way the endoplasmic reticulum in that the inner part of the microsome, that's similar to the lumen of the endoplasmic reticulum. And so the membrane here on the microsome is the same as the endoplasmic reticulum membrane. And so they knew that when you form these microsomes, that there's still ribosomes attached. The question was, are proteins that are destined for secretion, would they be found inside the lumen of the microsome? Meaning that in an intact cell, they had to enter into the lumen of the intact endoplasmic reticulum. So this is an experiment. It's an artificial test of the hypothesis: do secreted proteins have to go through the endoplasmic reticulum? That's the hypothesis. The experiment they designed was to homogenize, break up the cells, allow the endoplasmic reticulum to form microsomes, which they can kind of isolate, and then take those isolated microsomes and then do two experiments where they split them in half. So one half of the microsomes they treat with a protease, and the other is basically the same, but before they treat it with a protease, they add a detergent, and the detergent is able to puncture little holes into the microsomal membrane. And so when they do that detergent step, and then they add the protease, so the protease, what is that? It's an enzyme that cleans up other proteins, chews them to pieces. And so in this case, at the very end of this, we take this sample, both these samples, and we run them separately on sodium diacetyl sulfate polyacrylamide gel electrophoresis, the way of separating proteins. How many people here have ever learned about SDS page? Okay, good part of it, right? So it's a very, very common technique used in cell biology and biochemistry to measure levels of certain proteins. And so, in this case, when they added the detergent punctured holes in the membrane, and when they add the protease can get inside to the inner part of the microsome and chew up all the proteins that were there. In this case, because there was no detergent added, the microsomal membrane is still intact. The protease cannot get in. And therefore, when they take this sample and they run it on the SDS page, lots of proteins are seen. You can see bands in that gel. And here, you see no bands, because everything is being degraded. There's no intact proteins. And so that was the first proof that secreted proteins must first go through the endoplasmic reticulum. So it's just a proof of proof of principle that secreted proteins have to go through the endoplasmic reticulum. So remember, we need to break the cell apart. It's going to form microsomes. Microsomes are a representation of the endoplasmic reticulum. The membrane here on the microsome is the same as the membrane here. The inner part here is the same as the lumen of the endoplasmic reticulum. And the outside will be what you'd find on the outside membrane of the endoplasmic reticulum. Membranes don't allow proteins to go through, including proteases. So if you don't add a detergent, the protease cannot try to get in there and chew up all those secreted proteins. So they remain intact. In this case, we add a detergent, punctures little holes, in goes the protease, chews up everything. When we analyze these samples by SDS page and we stain for proteins, we see nothing here, and we see bands here because they're intact. That should make sense, I hope. So, that was hypothesis one. Secreted proteins had to go through the endoplasmic reticulum. They proved it. The next step was, well, are proteins made fully in the cytosol? And those big cumbersome proteins, are they then somehow inserted or moved into the endoplasmic reticulum? So in the first experiment, what they did is, this is what we call a cell-free or in vitro protein synthesis assay. So what you can do is take messenger RNA from a cell. There's ways that you can, I don't want to get too deep in the weeds. There's a way that you can artificially express the messenger RNA, and you can also have it translated. So it's an in vitro transcription translation system. So usually it's a certain purified lysate that contains all of the translational components found in the cell. So you're doing protein translation in a tube. This is not in the cell. So we introduce our messenger RNA, we introduce the protein ribosomes and translation factors and all that good stuff, and we allow the in vitro, in a tube, translation of those proteins. And then we take those artificially produced proteins and we mix them with microsomes that we purified from the cell. And then we look, using SDS page, to see, was there any incorporation of those proteins into the microsome? And the answer is no. So this showed that if we fully synthesize a protein, for example, in the cytosol, that fully synthesized protein cannot get into the endoplasmic reticulum. That's experiment one. Experiment two was very similar to this experiment one. The only difference is we put everything together. It's like a big gumbo soup. Put all the in vitro, the messenger RNA and in vitro transcription translation stuff together, and we also added microsomes all at the same time. And what happened is, now we can detect proteins in the lumen of the microsome. So it required the translational apparatus to be intact and associate with the microsomal membrane that would allow what we call co-translational translocation. So we're translating and transporting or translocating that emerging synthesized protein into the lumen of the eye. Remember, this is an artificial representation of the inside of the endoplasmic reticulum. So again, the key thing here is that this experiment demonstrated that translocation of secretories into microsomes is coupled to translation. So that was a major finding. And I'll mention just because it just showed here on here, it talks about a signal sequence. So when they looked at the proteins that were ultimately here, and they compared them, for example, proteins that were synthesized here, they found that these proteins were just a little bit smaller. There's like a little piece that would be removed. As we'll talk about in the next slide, that little piece is a signal sequence. And that signal sequence is really important for guiding this emerging protein to the membrane of the endoplasmic reticulum. Okay, so how do we put this all together? Can you give a little more detail about how that works? So as I mentioned, remember, if we go back to here, if you fully synthesize these proteins, and you don't have any microsomes, no ER is present, these proteins have a little bit extra at the immunoterminus, the very beginning of the protein. Whereas if we do this experiment and we mix everything together and allow translation and translocation to occur at the same time, when we look at the proteins here, you see that little red piece? It's not found. These proteins are a little bit smaller because the piece has been cleaved off. And that little red piece is a signal sequence, which we'll talk about in a minute. And it's going to be recognized by a protein called a signal recognition particle, or SRP. And there's a receptor for that, and that's found at the endoplasmic reticulum membrane. And it's going to help bring a whole complex over to this thing called the translocon, or protein channel. And finally, that signal sequence is going to be cleaved off. So I'm just introducing the steps right now, but the picture says 1,000 words, and that's what this is. So there's a lot of steps here, but just follow it, and it makes logical sense. So let's say we've got a messenger RNA. It's floating around in the cytosol. It's going to be encountered by a ribosome. The ribosome is going to recognize, hey, this is messenger RNA, and then start translating. So it'll start translating it. And as that emerging peptide, or emerging or nascent, we sometimes call it polypeptide, starts to emerge into the cytosol, the first number of amino acids that are synthesized at the amino terminus are going to be recognized, are what we call a signal sequence. And they, in turn, will be recognized by a signal recognition particle that's floating around. So it binds to this emerging polypeptide, which is still associated with the ribosome and the messenger RNA. So it's a big complex of RNA and protein. And that's going to guide it down to the endoplasmic reticulum membrane. So this yellow part, that's the cytosol. This is the air membrane. The blue part is the lumen, the inside of the endoplasmic reticulum. And so it will bind this whole complex via the signal recognition particle, SRP, will bind to the SRP receptor that's found at the ER membrane. So it has a portion that's facing into the cytosol, and that's going to bind to this big complex. And once it binds, it's also going to cause the recruitment of GTP. Remember we talked about that, of GTP can be used as an energy source? So when GTP binds to this complex, it can get hydrolyzed. It's broken down to GDP. And that provides enough energy to help dissociate this whole complex, dissociate the signal recognition particle from the ribosome and the receptor. And it also allows the ribosome to move over to this purple structure, which is a translocon. So translocon is like a little, I don't know, imagine a donut. And you've got the hole in the donut, but the donut can squeeze and constrict and block the hole in the middle, or it can relax. And so a series of events that happen here. The exact sequence does not matter. I'm going to just know exactly when does the GTP bind, when does it really, blah, 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 it doesn't matter. It's important to know the concept of what are the pieces that you need to have in play. I don't give a crap about what order. I've been doing this for like 18 years, and everybody gets all about what is the order of this. There is a certain order in that, we can go through. Protein has to be made, has to expose the signal sequence. That's going to be recognized by the SRP. That's going to guide it down to the ER membrane and bind to the SRP receptor. Now, GTP binds at this stage, and then it hydrolyzes. Now, does a hydrolysis, what is it important for? Well, it's probably important for now promoting the dissociation of the SRP from the SRP receptor. So now that free SRP can go out and bind and find another protein with a signal sequence. It's probably also important for providing some energy to kind of move this over. And whether or not it promotes the opening of the translocon channel or not, I don't know, I really don't care. What's important is that in order for this to get into the lumen of the ER, the translocon has to open up. And that's going to allow that protein, that emergent protein, to now move through the translocon, through the little donut hole, into the lumen of the endoplasmic reticulum. And as that moves through, when enough enters in and fully exposes the signal sequence, there's a protein called a signal peptidase. It recognizes, oh, that's a signal sequence, and blocks it off. So it's cleaved off. Translation still continues. So the protein still is getting translated, amino acids, beads put on the string, and it's pushing. The beads that are already on that string are getting pushed further into the lumen of the ER. And then finally, at the very end, you have the complete protein that emerges and it folds, the translocon closes, and the ribosome detaches. So I think what's important to understand are what are the things that we would need in order for, say, a secreted protein or protein that has to go to the lysosome. What are the things that it would need? It would need a signal sequence. It would need the requirement of SRP that functions. SRP would have to have a functional receptor at the ER membrane. GTP would be involved at some point. So you could, if there was no GTP, what would happen? Well, this would bind. You'd have this complex bound to the SRP receptor, but it would be kind of stuck there. It couldn't go any further. The GTP hydrolysis is important for dissociating the SRP from the receptor and ultimately moving this complex over to the translocon. And once that happens, it's going to open up the channel, and that's going to allow that emerging, newly synthesized protein to go through the donor hole into the lumen of the endoplasmic reticulum. What's the next step? The signal peptidase has to be activated. It will recognize that signal sequence, and it's going to lock it off. Translation continues to occur, and ultimately you have a fully translated protein, emerges into the lumen of the ER, translocon closes, the ribosome detaches, and this all starts over again. So what happens to proteins in the ER? Many things. They can be further, we talked about the SRP, there's a signal peptidase and cleavage. There's other types of proteins that can cleave in the ER. There's things that can be added on, including carbohydrate groups, that's called glycosylation. There's the formation of disulfide bonds, so that occurs through cysteine residues. They have sulfur atoms. Two sulfur atoms can form a covalent bond called a disulfide bond. And then also it's important for these proteins to be folded properly. So glycosylation in the ER, I'm not going to get into it in great detail, but it's important to note that there's this pre-manufactured, synthesized 14 residual oligosaccharide. Oligo means many, saccharide means sugar, so many sugars. There's different types, mannose and glucose, galactose, I don't know, a bunch of other things. And so it's anchored onto the inner membrane via a dolichol. It's a membrane-anchored phospholipid. And it can be, when an emergent protein is coming into the lumen of the ER, it can be recognized by an enzyme called an oligosaccharyl transferase. And it can transfer this branched sugar structure onto amino acid, in particular asparagine. And so once this happens, so you have this big branched sugar structure stuck on your protein, it can then undergo further removal. So you have generally what happens is you put on this big branched sugar and then you cleave some of those sugars off. So they're called, the things that take the sugars off are called glycosidases. And so glycosylation is really why cells put these big sticky sugars on proteins. Well, it does a lot of things. It can affect how proteins are folded, so I'll talk about that in a minute. It can make the proteins more stable. It can also make them more sticky. So remember, when we were talking about trypsin and when we passage cells or breaking tissues apart, the trypsin cleaves off the extracellular matrix proteins. Those are sticky proteins. Part of what makes them sticky is they tend to be heavily glycosylated. The sugars themselves actually confer adhesion. So cells that proteins that are involved in cellulation would have to go through this process starting in the endoplasmic reticulum. And we'll talk maybe a little bit later about how some sugars are added or taken off. So the glycosidases take off the extra sugars. The enzyme that's important for putting them on, particularly the first step is the oligosaccharyl transferase. So protein folding in the ER, it's a very massive topic, and I don't think we just briefly touch upon it. It's really important for proteins to fold properly when they go into the ER. And quite frequently what happens is proteins are emerging into the ER. They might have a predisposition. There's a long series of hydrophobic amino acids. Normally those hydrophobic amino acids were found in the center of the protein once it's folded. But when they were emerging out, they could start to maybe interact inappropriately or with other proteins that are close by in the hydrophobic regions. And so we need to prevent that inappropriate misfolding of the protein. So what can happen is as the protein emerges, and let's say it's got a stretch of hydrophobic regions that could cause misfolding as the protein is starting to emerge, you have these chaperone proteins. So one is BIP, and there's some other ones called calnexin and calreticulin. We'll talk about in a second. So what happens is, as that protein is emerging, the oligosaccharide may recognize, oh, this is a hydrophobic region, or here's a part of the protein that's starting to misfold. How it senses that is a whole other story. And so it sticks on this complex carbohydrate structure, and then that causes recruitment of the BIP, chaperone protein. And the chaperone is exactly like that. It grabs a hand of that protein and says, no, you're going to go this way. We're not going somewhere else. You need to behave, you need to move in this direction and fold in this direction. And so as the protein continues to emerge, the BIP proteins hold on to usually hydrophobic regions, preventing them from associating inappropriately with other hydrophobic regions in the protein, or even another adjacent protein that happens to be floated by. Calnexin and calreticulin do, so BIP is kind of free-floating in the lumen. Calnexin is also, these particular proteins actually bind to the carbohydrates themselves. So they recognize the carbohydrates, they have carbohydrate recognition domains. And so they recognize, oh, here's a big sticky piece of oligosaccharide stuck on this protein. I'm going to hold on to it as it continues to be synthesized to prevent any mislocalization, or misfolding, I should say, of the protein. So calnexin happens to be associated with the inner membrane, calreticulin floats in the lumen of the ER, but both of them bind to the carbohydrates, not the actual amino acids. The other protein that, let's see, what else are we going to talk about? Okay, PDI, protein disulfide isomerase. So as that protein is emerging, and it has, for example, a cysteine residue that has a sulfur, it may want to form a bond with another cysteine residue over here, but it's not supposed to. And so what happens is it forms what we call a mixed disulfide. So here's the polypeptide emerging, that cysteine residue with the sulfur atom is present, and protein disulfide isomerase also has a cysteine with the sulfur, and it forms a temporary covalent linkage. That's the disulfide bond. And it kind of holds on to that protein as it emerges more, and then there's a mechanism to break this disulfide bond later. So it forms a transient temporary disulfide bond to prevent it from forming a disulfide bond with another part of the protein or another protein. So eventually it kind of holds off on, it kind of guides this sulfur atom along until enough of the protein is made. It's like, okay, I can now pass you on to the proper cysteine residue elsewhere in the protein to form these stable disulfide links. And those disulfide links are really what helps stabilize that particular protein, particularly secreted proteins. So if you look at immunoglobulins or antibodies, they have a lot of these disulfide bonds, and that's what gives them this incredible structure that can withstand a lot of beating when they're secreted outside of cells. Antibodies are very stable, in part because they form these complex disulfide bonds with stables. They're covalent, not ionic, literally covalent bonds that stabilize the structure. So ultimately, as the protein emerges fully, it will fold properly, it will have appropriate disulfide bonds, particularly if it's secreted, and it will also likely have glycosylated asparaginase. And hopefully these proteins that assist in the folding allow it to fold its full structure, and then it can form, for example, sometimes proteins will form with another protein. Mm-hmm.
So as I understand it, these chaperone proteins are not unique for each protein. So how do they know where the right sulfur is, for instance, like when to let go?
Okay, so good question. How do the chaperones know which protein they need to bind to? Are they unique to each protein? No, not necessarily. A chaperone can bind to many different proteins, but usually there's a signal. So the chaperone protein, for example, BIP, or I should say calnexin and calreticulin, they know to bind to that protein because it has that branched sugar added to it. It's the recognition of the sugar is what promotes its binding. And it holds on to that glycosylated portion of the protein until it's fully formed, and then it can release it. In terms of protein disulfide isomerase, some of this is dependent upon recognition sequences in close proximity to the sulfur. I'm not going to really get into too much detail, but there's ways, they don't just willy-nilly grab onto anything. There's usually sequences found within the protein that are recognition sequences that promote the association of the chaperone proteins. And ultimately, we want these proteins to associate. They can form complex structures with other proteins or another version of itself to form a dimer or a trimer. And so once they're properly folded, they can be transported from the endoplasmic reticulum to the Golgi, which we'll talk about in the next lecture. Okay, so I don't know if I'll get to nuclear, but at least we can do the mitochondria. So mitochondria are mitochondrial proteins. So say, for example, a Krebs cycle protein. How is it going to get A from the cytosol into the matrix? How does it know when it's made in the cytosol to go to the matrix? And so, as I mentioned, there's these targeting sequences. So mitochondrial matrix targeting sequence is about 20 to 50 amino acids, usually at the amino terminus, although there's some exceptions to the rule. And they have both hydrophobic, basic, and hydroxylated amino acids. So that means they have hydrophobic, doesn't like water, and hydrophilic, loves water. So they have this combination of both neutral or hydrophobic sequence and charged sequence is what we call amphipathic. And it basically means it has both hydrophobic and hydrophilic or charged sequences. And so that targeting sequence is helps direct it towards what we call these certain groups of proteins called TOMs, translocation of the outer membrane. Now, before it gets there, there's also chaperone proteins that are found in the cytosol. And there's a whole group of them called heat shock proteins. So cytosolic heat shock protein will bind to this particular protein and prevent it from misfolding in the cytosol. And also, with association of a bunch of other proteins, and partly dependent on this targeting sequence, can also help guide it towards this TOM. So there's a whole bunch of TOMs, and I'm going to talk about TIM in a minute. I don't really care about numbers. Numbers don't really matter. 22, 47, 58, 101, I don't care. It's just important to know that TOM, there's a number of translocation of the outer membrane. That's what TOM stands for. And that one of them can act as a receptor. So it can bind to the signal sequence and then guide it over to another TOM. And this one functions very much like the translocation of the endoplasmic reticulum membrane. It's a channel, so it can open up and allow the movement of that protein into the inner membrane space. Now, usually what happens is that cristae, remember the undulating inner membrane of the mitochondria, so it goes back and forth. When you have an association between the inner membrane and the outer membrane, where they're close together, quite frequently what we'll see is a clustering of TOM proteins, translocons of the outer membrane, associating with their counterpart, which is TIM, translocon of the inner membrane. So TOM and TIM get close to one another, their translocons their channels can open, and so this allows the movement of that protein across the outer membrane, past this little tiny inner membrane space, and then moves through the translocon of the inner membrane. And that will start to enter into the matrix. And there's a matrix HSP. It's a mitochondrial localized HSP, which helps pull it in and prevent it from misfolding at the same time. This requires energy, ATPs. Energy is required at both steps to prevent misfolding and also guiding. and also pulling in that protein here into the matrix, and also preventing it from misfolding. And ultimately, there's a protease which will cleave off the targeting signals. Okay, so we'll kind of touch back on this on Wednesday to finish it up, and then move on to other stuff.
Advice is don't miss lecture if you can avoid it. If you do make miss lecture, then obviously try to see if you have a kind of class that helps. But in terms of studying, I think that curious. Right now, it's obviously like last class is September. So when you have your lecture notes, you find out you do it. When it comes to finding you have no lecture notes, that's true. I recognize don't just continue to go through your notes over and over again. It's to read through your notes, close up the book, and make summary pages, sort of, what do they call it, mind maps of, hey, what are all the steps that are involved in co-translational translation? Write it out. You can do that and not have to yourself notes, but you know. So kind of don't just continue to read your notes over again and read your textbook over again. Close it up and make summary notes for yourself. Or if you have explained this.
Love my boys commenting.